Next chapter, chapter 4, we'll be talking about the tissue level of organization. Next level up from cellular level. For a lot of you, this will be new information. We're going to be talking about the 4 different types of tissues. We're going to do a brief overview of each set. We'll go through a little more in depth as we go through the semester. In fact, in some of the tissues, we'll be going through numerous chapters, as many as 6 or 7. We'll be going through that as we go further on. So the purpose of this chapter is we'll be able to identify the different types of tissues and what kind of organs that they will comprise. We'll talk about how tissues are held together. We'll compare and contrast the 4 types of tissues, in this case epithelial, connective, muscular, and nervous tissue. And we'll talk about different types of membranes, and finally we'll delve into tissue repair. For those of you who are going to get into the health fields, this is a very important topic, as not every type of tissue will actually repair itself a little bit differently than others. Some need some additional assistance, such as surgery or medication, and some can actually repair themselves fairly rapidly. So the 4 main tissue types, again, are epithelial, connective, muscular, and nervous. Epithelial tissue, as we'll talk about later on, can come in different forms, but for the most part, they line either an outside surface or the surface that faces a lumen or an opening. Connective tissue is a very, very broad category, where basically the number of cells is outmatched by a number of ground substance or surrounding fluid. Muscular, most of you should be very familiar with. There's 3 types of muscle tissue we'll talk about: cardiac, smooth, and skeletal. And finally, nervous tissue, which we'll do a very brief overview here in this particular chapter. We'll go more in depth in the second half of the semester. So first, let's talk about different types of cell junctions. So especially with epithelial tissue, muscle tissue, and some forms of nervous tissue, these cells are bound together by different types of cell junctions. Now this animation here is a very nice, very brief overview of all different intercellular junctions. We'll talk about those in the next couple of slides. So those of you who would like to click through that through the normal PowerPoint presentation, then come back, at least have a better idea, or at least reinforce some locations that you find these junctions in certain types of tissues. The first types of junctions we'll talk about are tight junctions. We find these in particular binding together the epithelial cells that may be contained in the stomach, or through the gastrointestinal tract, or even in the genitourinary tract. For the most part, these are there to make sure no fluids will actually pass through the cells and cause any further damage to the basal membrane or any other underlying cells. In this case, you see 2 adjacent plasma membranes. You'll see transmembrane proteins. They'll essentially fuse the 2 membranes together and form a very tight knit type of space between the 2 cells to prevent any kind of fluid from passing through. There still is some intercellular space, which allows certain gases and certain types of products and waste to pass through each individual cell. But in this case, for the stomach, let's say, this prevents hydrochloric acid from passing through and damaging the cells found deeper, closer to the basal membrane, or as I said before, to deeper types of tissues. Next, we have adherence junctions. With this, you'll have the actin molecule, which will be forming what's called an adhesion belt. This will be forming between adjacent plasma membranes, so it's not quite as tight as the tight junctions will be. You'll have the transmembrane protein, the glycoproteins, cahedrin, that will basically form like a meshwork, almost like a zipper-like quality, binding the 2 cells together. This will have a little more of an intercellular space, a little more consistent as it goes throughout the plasma membranes. You find this in where you find areas of high temperance and also high tension. So in this case, along the esophagus, along certain areas of the gastrointestinal tract, we have smooth muscles surrounding. So it's these adherence junctions that prevents the cells from tearing apart as the muscles contract over top. Next, we have desmosomes, which have a slightly different configuration than adherence junctions. They'll still have the transmembrane glycoproteins, cahedrin. They'll bind the desmosomes together. It's almost like stamped buttons on either side of the plasma membrane. This is another way of anchoring cells together, except this one uses the intermediate filaments, if you remember those from the last chapter, in this case keratin, instead of the actin filaments. This is also used to help bind cells together, most likely at the base of the membrane, sometimes higher up. This will also allow some intercellular space in between the adjacent plasma membranes, and also is involved in areas of high torsion. You'll find these binding epithelial cells found along the mouth, along the throat, and also along the gastrointestinal tract, as well as the urethra and a few other key spots. But again, these are mainly used to help bind adjacent cells together to prevent them from tearing apart due to mechanical stress. Now, a special type of desmosome is what's known as a hemidesmosome. Now, this one is basically situated the same way as a desmosome is, except instead of attaching two cells together, is going to anchor the bottom layer or the basal layer of cells to a basement membrane. In particular, you see this with the basal layer of epithelial cells, which we'll talk about a little later on. So it's basically one half of desmosome. You still have the integrin glycoproteins anchoring the basement membrane to the cell, and also the keratin filaments found on the other, or the intracellular half of the desmosome, basically holding the cell to the basement membrane. And finally, we have the gap junctions. We find these in particular between cardiac muscle cells and also a few nerve cells. So this one, it'll form connexons or tunnels, which basically allow ions to pass through very, very quickly. It's this kind of ion flow that allows adjacent cardiac muscle cells to contract. So very simply, one cardiac muscle cell contracts, the next one down the line will contract with it. This allows the heart to pump as one unit in particular. It does allow a little bit of a gap, so this way you can have contraction through the atrial and the atrial areas as well as the ventricles, so not all contracting at the same time. It allows a little bit of a delay as ions will pass through the adjacent gap junctions. Allows the muscle to contract. We often see these in some form of small muscles as well with a lot of contraction, but we also see gap junctions in some neurons, which we'll get to later on. Most neurons will fire off through gaps to the synapses, but some actually connect through gap junctions. We'll get through those when we get into some of the nervous system chapters. Now, one little exercise I want to introduce to you is very simply, we're looking at different types of slides. To be able to differentiate one particular type of tissue from another, or one subcategory of tissue from another, is to basically look at the common as well as the contrasting characteristics of each one. So this one in particular, we have epithelial tissue on the left-hand side, we have connective tissue on the right-hand side. So one of the defining features of epithelial tissue is going to be very little intracellular space. You're going to have the epithelial cells bound together by tight junctions, adherence junctions, as well as desmosomes and so on and so forth. And as you can see on the left-hand slide here, there's multiple nuclei, dense, tightly packed cells, very little extracellular matrix. Conversely, on the connective tissue side, you'll find this in almost all different connective tissues. In fact, this is how we will classify connective tissue. You'll see very few cells compared to a very large amount of extracellular matrix. And that kind of matrix can be anything from bone to blood to cartilage, as well as a few other things. We'll go through each one as we get through the chapter. So first up, epithelial tissue. So general features of epithelial tissue, they are going to be arranged in sheets, sometimes one single layer, sometimes in multiple layers. They are densely packed as they are stitched together by tight junctions. The epithelial cells will always attach to a basement membrane. The basement membrane is usually going to be formed of connective tissue. Now, epithelial tissue is avascular, meaning that there's not a good blood supply, but does have a fairly good nerve supply. Now, the vascularity comes from the basement membrane in particular, and will diffuse usually from the deepest portion to the more superficial portion of the epithelial cells. And especially in areas of high friction, usually in the mouth, usually in areas of urination or in defecation, you're going to have a high rate of mitosis as you're constantly trying to replenish the epithelial tissue. In this case, skin in particular will actually regenerate within about 4 to 6 weeks. Now, another feature of all epithelial tissue is you always have an apical surface, which will face either the outside environment or a lumen. And you also have a basal surface, which will contact the basement membrane. So in this case here, we see a single layer of epithelial cells. We see the apical free surface at the top of the slide here. You have the lateral surface of each of the plasma membranes adjacent to each other, stitched together by tight junctions and adherence junctions. And you see that the basement membrane is going to attach at the basal surface of the epithelial cells by hemidesmosomes. In this case, the basement membrane is going to be split into what's called the basal lamina and the reticular lamina. The basal lamina is going to be the most superficial, will attach directly to the epithelial cells, whereas the reticular lamina will actually connect to the connective tissue at a deeper level. And this is where you'll find your blood vessels and also the nerves as well. The nerves will actually extend into the basement membrane just at the base of what we call the basal layer of the epithelial surface. Of the epithelial cells. You'll find these for touch, temperature, itch, as well as pain, which we'll get into in the integumentary system chapter. So epithelial tissue can be classified different ways. It can be classified by the arrangement of the layers. So in this case, simple is just one single layer, so one layer thick. You can have what's called pseudostratified, where you can have the appearance of the nuclei being different positions, but in essence, there's only one particular cell, one layer thick. But the way the epithelial cells will twist and bend around one another, the nuclei will get shunted to different spots. And we'll talk about different types of epithelial tissues that are pseudostratified in the next chapter. And finally, for the arrangement of layers, you can have stratified, which is multiple layers of epithelial cells. In this case, you see multiple stacks on the far right-hand corner. But notice the difference between the simple and the stratified and the pseudostratified. The simple and stratified, all the nuclei, for the most part, will line up in one particular plane, whereas pseudostratified, due to the twisting and bending nature of the individual cells, has the appearance of having multinucleation, when in essence it's only a single nucleus, but it's being pushed into different areas to the path of least resistance. Besides the arrangement, we also look at different cell shapes. So in this case, we see what's called squamous, which are very flat. These are usually found lining lung tissue as well as capillaries. They're very good for allowing gases to pass through the epithelial layer, in this case oxygen and carbon dioxide. You'll see cuboidal, which is exactly the way it sounds. They're shaped like cubes. And finally have columnar, which will be a higher height than is with width. But for the most part, columnar will have most of their nuclei all along the basement membrane. The last one not shown here is called transitional. We only find these in the urinary bladder and certain areas of the urethra. These will start out as cuboidal, but have the capability of stretching. They'll also be in a stratified position. In the next slide, it will link into Wiley Plus, which will give you a demonstration of how each one of the different types of arrangement layers, as well as the cell shapes, and what epithelial cells or what epithelial tissue you'll find them in. So again, table 4.1 found in your textbook. I find the textbook, as well as Wiley Plus, is a very nice representation of different types of cells you'll find throughout the epithelial tissue, throughout the human body. And we'll talk about, again, more in depth of what each individual epithelial tissue does. For the most part, for this chapter, just know what kind of epithelial tissue, how they're classified either by their shape or by how they're layered, or if they're transitional. And here's a link through the Wiley Plus. This one's a little bit different than the animations. This one is not going to be voice driven. This one is a click through. So I hope you'll click through this particular link and go through each individual slide. The slides you're going to see will be testable. When we do quizzes in the next week or two, you'll be answering questions based off the slides you'll see both in this particular link and also in the textbook. So again, a quick little chart here, just showing different types of naming combinations. Simple squamous, again, you'll find mostly in capillary beds as well as areas around the alveolar areas of the lung. Cuboidal, simple cuboidal, normally you'll find these as secretory, secreting hormones and other types of products. Simple columnar, you may find these in different types of areas of the gastrointestinal tract, as well as different areas of the different types of glands. Pseudostratified, you may find these in the stomach or the small and large intestine. Stratified squamous, you find these usually in the mouth, on layers of the skin, on the heel of the foot, and we'll talk about different types of layers when we look at the outside skin in particular. Stratified cuboidal, again, see these mostly in glandular tissue, and also see columnar stratified tissue. You also find these in glandular tissue as well. And transitional, we find this, since it's very elastic, very stretchy, you find this in urinary bladder and the urethra. Now, one special feature of epithelium is it has the capacity to secrete. So in this case, we see glandular epithelium, and we see two different types. We see both endocrine and exocrine. And we classify each one by very simply, the endocrine will actually dump its products into a blood vessel. In this case, it may actually be the entire cell or a certain portion of the cell, or possibly just vesicles just being exported out into the blood vessel in particular. And here you see endocrine gland, thyroid gland, which will dump out thyroid hormone directly into the blood vessel. Exocrine glands, by differentiation, will dump their products into a free surface. In this case, your sweat glands, your sebaceous glands for different types of oils for the skin, ceruminous glands, which will secrete earwax into the ear canal. So here we see an example of the sweat glands secreting their their products into the lumen, or the opening of the sweat gland, to be dumped out to the skin surface. Now, aside from being classified as either exocrine or endocrine, we also have structural classification of glandular epithelium. We do have quite a few here. There's only going to be a few that I'm going to point out to you that's going to be testable. The others will be pretty much covered more so in HAP 2. Very simply, we look at the structure of the glands. We look at either unicellular, which means it's a single cell secreting its products, or multicellular, which very simply means it's comprised of numerous cells that form a distinctive structure or an organ. So in this case, your sweat glands, oil glands, and salivary glands will be multicellular. So here we see different types, different configurations of different types of glands. There's only two in particular I want you to pay attention to. Is letter C, simple coiled tubular, be able to identify this from the slide, and also letter E, simple branched acinar. So again, letter C, simple coiled tubular, and letter E, simple branched acinar. I want you to be able to pick these out from the slide if at all possible. The others, the simple tubular, the branched tubular, the simple acinar, compound tubular, compound acinar, and compound tubular acinar, we cover more so in HAP 2. Now, of the multicellular exocrine glands, only two, like I said before, are going to be important for this particular chapter, and especially we get into the skin, and that's going to be the simple coiled tubular, which comprise your sweat glands, and also the simple branches acinar, which will include your sebaceous glands. This will form the oils onto the skin and also helps maintain proper structure for your hair as well. The other glands I want you to just be aware of, and just found that they're going to be multicellular as well as exocrine. So the simple tubular, found in the large intestine, simple branch tubular, the gastric glands, the simple acinar, the glands of the penile urethra, the compound tubular, found in the bulbar urethral gland, the compound acinar, the mammary glands, and the compound tubular acinar glands of the pancreas. All these will be found more so in the second part of human anatomy physiology, which will deal with genitourinary as well as gastrointestinal. Now we also look at different functions of glands. In this case, functional classification of glandular epithelium. We break them down into three types. There's merocrine, where very simply secretory vesicles will dump out its products in a secretion, such as salivary gland. So in this case, it's dumping it out into the free surface, in this case, the mouth. You also have apocrine, where a portion of the cell gets pinched off. In this case, there's a form of cytokinesis that pinches off a portion of the cell, and that in itself is a secretion. In this case, the mammary gland does this type of secretion of glands. And finally, you can have a holocrine, where very simply the entire cell dies off and becomes the secretion itself. So in this case, for sebaceous or oil glands, the entire or the whole cell, hence the name holocrine, becomes the entire secretion. So it'll mitocondrially divide, and then very simply it will die or through autopsis, and basically itself would be the secretion. Next, we'll talk about connective tissue. Connective tissue is probably the broadest category that we have here. You'll see very simply that compared to epithelial tissue, there will be less cells that you'll see versus epithelial tissue, but there will be an abundant amount of extracellular matrix, which in this case may be fluid or may be hard tissue. The general features of connective tissue includes, again, as I mentioned before, very few cells compared to an abundance of extracellular matrix. The cells do not cover or line any particular type of organ that faces an outside surface. So you will not find any connective tissue that will line organs. You find mostly epithelial tissue will line those. You won't find anything exposed to the outside surface. Usually those will be comprised of epithelial tissue or different types of membranes, which we'll get to in a few minutes. By comparison, epithelial tissue is highly avascular. However, most connective tissue, not all, is highly vascularized. It has a very abundant nerve supply as well. The main exception will be tendon and cartilage. We'll get into that a little later on into the joint chapters, especially cartilage found in the knee, also along the spinal column and an ear. It's very avascular, which makes it very difficult for that tissue to repair. We'll talk about that later on in the chapter. In this slide, we see some typical cells we'll find in connective tissue, although some, such as in bone as well as cartilage, you'll find osteoblast and osteochondroblast, respectively, in those particular connective tissues. This is still representative of a typical connective tissue area, like loose areolar or dense irregular connective tissue. Fibroblasts are the cells you see in the middle there. You see those secreting fibers for ground substance as well as collagen, reticular fibers, and elastic fibers. The collagen fibers are there to give more tensile strength so it doesn't rip the area apart. You find these around the areas such as the lung, as well as areas around the kidneys, the spleen, so on and so forth. You also find other types of fibers like elastic fibers. These will help the area stretch. So in particular, when you see the lungs expand and contract, the elastic fibers allow that to happen. And finally, have what's not pictured here are reticular fibers. This basically provides a stroma or framework for all the individual cells. You'll see interspersed throughout there as well different types of white blood cells. You'll see large macrophages, which are basically there for patrolling for bacteria and for cell debris to consume by phagocytosis. You'll find plasma cells from the B lymphocytes. These will be important for secreting antibodies to neutralize any kind of viruses or any other form of substances. You have mast cells that usually around or linger around the blood capillaries and other blood vessels, which will produce histamine to dilate small blood vessels during inflammation. Some of our mast cells are a little too eager and cause a massive inflammatory response. This is where a lot of allergies will stem from. Other white blood cells will include neutrophils and eosinophils, which are very important for fighting any kind of infection due to microbes or any kind of parasitic infection, respectively. And finally, we see adipocytes, which are your fat cells. Depending on the actual area, the adipocytes may be sparse or it may be filling the entire region with lipids for both for energy as well as for cushioning at the same time. So the extracellular matrix is located between the spaces between the connective tissue cells. Usually the matrix is composed of fibers and ground substance. In some areas, the ground substance is hard, as in bone, or it could be semi-fluid, as in cartilage, or it could be very fluid, found in blood and in some loose areolar tissue. As I mentioned before, different types of fibers that we find in the extracellular matrix. We find collagen fibers, which are flexible but don't stretch. So this gives you some tensile strength in the area. Collagen fibers are also the most abundant protein found in the body. We have elastic fibers that allow the area to stretch up to 150%. And finally, reticular fibers. This forms the framework for the actual tissue itself and allows the different capillaries, veins, blood vessels interspersed with adipocytes to hold on to that particular framework and maintain proper structure. So classification of connective tissue comes in two forms. We have embryonic and mature. For embryonic, we have the mesenchyme and mucosal. Mesenchyme is going to be the starting tissue before it differentiates into different types of mature connective tissue, as we'll see in a couple slides. Now, out of the mature tissue, we're going to find loose, dense, cartilage, bone, and blood. When we get into lab and also into the next slide here, Wiley Plus has a very excellent representation or animation for each of these different types of mature connective tissue. I encourage everyone to check both the real-life slides, the scanning electron microscope, as well as the light microscope, and also the artist representations of different types of tissues here. These will be material to be used for quizzes and or the exam. In particular, quizzes will be done by PowerPoint slides. You'll be asked to identify each individual connective tissue slide. I'll do my best in lab to give you different ideas of what they look like. You'll see both best representations from the textbook and then also slides we have in lab with looking through a light microscope. And here's your link for the connective tissues through Wiley Plus. This is a click-through slideshow mode. It's not a true animation with any kind of voiceover. You go at your own pace, and pretty much all the information you'll get from slide to slide is located within the textbook at the same time. And whenever we talk about connective tissue, we always come up upon the subject of cartilage. Cartilage is made up of three different types of molecules in differing amounts depending on the cartilage. Three main types being hyaline cartilage, which you find at different surfaces of each of the joints, which we'll talk about later in the joint chapter 9, articulations. You'll also find it in elastic cartilage found in the ear and also the epiglottis, and also in fiber cartilage found in the discs of the spine, as well as the menisci and some other key areas throughout the body. So basically the four joint cartilage proteoglycans are glucosamine sulfate, hyaluronic acid, chondroitin 4, and chondroitin 6 sulfate. Some of these can be supplanted by or supported by nutritional supplements, although with varying degrees of success, which we'll talk about later on. Some people actually will get these injected through either Synvisc or Orthovisc. When we get into the joint chapter, we'll talk about joint repair in particular, both from a exercise as well as a dietary or even a surgical standpoint. But these four joint cartilage proteoglycans are very important. It's important to get these into the diet as well as to maintain the joint integrity that you have already. Now, as I mentioned before, most connective tissue is highly vascularized with a few exceptions, cartilage being the major exception. In cartilage growth and repair, we have two types of growth, either interstitial, which means it goes from within and grows out toward the periphery, or appositional, where very simply the outer surface is very prolific as far as mitosis goes, as well as secretion of ground substance, and the outer edge starts to grow further away from the deepest part of the cartilage. Unfortunately, repair is very poor due to avascularization. Nutrients and waste have to pass through the tissue, and as I mentioned before, cartilage in particular is very almost gel-like, so it's very tough for anything to kind of pass through. The text recommends surgical repair, but current research and also current practice suggests otherwise. Most of the patients that I've sent out for evaluation for any type of surgery, whether it be to repair cartilage and or tendon and or ligaments, most orthopedic surgeons are loath to open someone up for simple tears, which we'll get into later on. But what they've found is they cause more damage going in, and then what they'd rather do is either modify exercise and or diet, or go through physical therapy to at least kind of load up on certain muscles and joints and try and repair things from within instead of trying to repair things from without. And let's talk about membranes. Membranes are very important in particular in that they're going to line different types of epithelial areas as well as synovial areas. The difference being the epithelial areas will line either an outside surface or it may line the edge of an organ, whereas synovial membranes are going to be found at the edges of each of the joints. So the membranes for the epithelial include mucus, which will secrete mucus, serous membranes will secrete serous fluids, and cutaneous membranes, which include the skin and also the areas around the ear, the sweat glands, and also the sebaceous glands found in the skin as well. Synovial membranes will secrete synovial fluid. We'll talk about that in a further chapter. Both slides will show up here in the next slide or two. And again, this is also material for any kind of quiz we may have where we're identifying structures. So here we see a letter A, a mucous membrane. So very simply, we have special goblet cells that will secrete mucus here. As you see, it secretes it to the apical surface of the epithelium. So this allows increased absorption for certain types of nutrients. Also allows for passage of gases if we're talking about the respiratory tract, as you see in the trachea. However, in the lungs themselves, you have a different type of membrane called the serous membrane. This is where, if you remember, the pleural cavities as well as the pericardial cavity found around the heart. The serous membrane is there to allow for lubrication of two body parts sliding upon one another. And obviously, with the lungs expanding, contracting, as well as the heart pumping, you want some kind of lubrication. Otherwise, you get what's called a dry rub, which is a very painful type of condition, where very simply the serous membrane lacks the serous fluid, and basically the two body parts start to rub against each other, cause friction, and start to destroy the tissue. So very simply, both of these will be epithelial tissue, both the mucous membrane and serous membrane. But you see that the configuration is a little bit different. You see columnar or even pseudostratified columnar cells with the mucous membrane interspersed with goblet cells, whereas the serous membrane is all flat squamous cells. It looks like a single layer here and has a single layer called mesothelum that will connect to the areolar connective tissue. You have the skin, which is the cutaneous membrane, which is going to be comprised of the epidermis, the dermis, and also the subcutaneous layer, which is the fat layer. We'll talk more about that in the next chapter. And the last and final type of membrane, which is not an epithelial membrane, it's actually a synovial membrane. You see the purple synoviocytes. This is going to be the leading edge in the synovial cavity. So this is actually a finger joint here. This is known as the synovial joint. The synovial membrane is going to surround both ends of the bone. It's going to be self-contained. It's going to secrete synovial fluid, which again is to help for nutrient as well as waste transport back and forth for the actual cartilage, and also provides a cushion for the joint itself. And let's talk about muscle tissue. So most muscle tissue that you've heard of will include the skeletal, the smooth, and also the cardiac. So basic muscle tissue will consist of different fibers, usually a combination of actin and myosin are the two primary contractile tissues. But there's also quite a few support level proteins that help maintain the actual muscle itself, the contractile tissue, as well as overstretching and so on and so forth. We'll get into more of that in the muscle chapter, a few chapters down the line. A secondary thing that it does too is help maintain posture, also produce heat. And one thing that most people don't think of, it also helps push blood through the venous system. If you remember, we have arterial pressure in the arterial system, but in the venous system is more of a reservoir. Especially when you look at people that are sedentary or people that are elderly, They tend to have some fluid buildup, usually in the extremities, in the hands or the feet. The first thing we ask them to do is simply just move the foot and move the hands around, try and pump that blood back through the venous system and back towards the heart to be redistributed throughout the body. So again, 3 types of muscle tissue include skeletal muscle, which we'll get into a little more. This is voluntary. The 2 others include cardiac and smooth. These are both involuntary for muscle contraction. So this is a good overview for muscle tissue. It'll go through different types of cells, in which you'll see under a microscope. So very simply, you'll see the interspersed patterns of actin and myosin with multinucleated cells all throughout the skeletal muscle. You'll see special intercalated discs. This is where the connexons, or the connexon junctions that we looked at, or the gap junctions for the cardiac muscle, and then you'll see spindle-like type cells down in the smooth muscle, very like a snail or a earthworm type of movement. And finally, come under nervous tissue. For the purpose of this chapter, for nervous tissue, we're only going to look at 2 different types of cells, and pretty easy to distinguish. Under a microscope, if you look at a neuron, neurons tend to have a trapezoidal or even a triangular cell body known as the soma. Depending on the slide, you may have a good look at the axon hillock as well as the long axon. You may see some dendrites as well sprouting off with the soma. The neuroglia will include all the different support level cells for the neuron to help protect through phagocytosis, or they may actually help with nutrient flow for the neurons themselves. They tend to be significantly smaller than the neuron itself. So very simply, the function of a neuron, it will carry both sensory and or motor information and also perform integrated functions. We'll dedicate an entire chapter to that type of physiology. And this is a quick little overview of nervous tissue. I believe there's only 1 or 2 slides here. Pretty self-explanatory, and the slides that we'll see in the lab as well will help support what you see on this particular animation. So let's talk about tissue repair, aging, and disorders. We're going to go through some very broad strokes through each one of these here. Tissue repair can happen in different ways depending on the tissue involved. Naturally, tissue repair is the process that's going to replace either worn out, damaged, or dead cells, or any kind of damaged fibers found within the cells. Epithelial cells, very simply, will be replaced by mitotic division of either stem cells or undifferentiated cells, and usually will move from the basal layer and move itself up towards the apical layer. And it's relatively rapid. You can replenish your skin in roughly 4 to 6 weeks. Connective tissues, some have a very strong ability to repair themselves, some not so. As I mentioned before, typically your cartilage tissue does not tend to repair very easily. Strangely enough, bone actually is very well vascularized, even though for most of us it takes a while to repair a fracture. It does repair more easily than does cartilage. However, muscle cells in particular can perform limited types of repair. Once we have fully mature muscle cells, we don't gain any more. Once they stop, they'll stop mitocondrially dividing once they fuse together, which we'll get into later on. As far as the skeletal muscle cells and also the cardiac, smooth muscle does have some form of limited repair, although it is relatively slow. However, muscle cells themselves can actually grow, in particular by increasing the number of fibers found within. So naturally, all repair is not necessarily a bad thing. Some muscle repair just from working out, whether it be from cardiovascular and or strength training, you're basically tearing up proteins or tearing up fibers within cells in the hopes that the body will lay down more tissue in those areas and therefore grow that particular tissue. Now, nervous cells in particular, for the most part, has limited repair under certain conditions, although some areas of the brain under current research over the last few years has noted that some neurons can replenish themselves. We'll get into that later on. We get into the brain and the spinal cord. Anytime you have any kind of filler tissue, it is known as fibrosis. This is the formation of scar tissue, which we generally see within the different types of connective tissue. In particular, you'll see scar tissue, which is a very random type thing found in muscle. You'll find it in cartilage. You'll find also in the epithelial layers as well. We'll investigate those a little more thoroughly as we get into each individual chapter. It should come as no surprise that as we age, our ability to repair ourselves starts to diminish. Naturally, younger bodies will have better way of replenishing themselves because we have better nutritional state, better blood supply, and also better metabolic rate. Unfortunately, aging will slow the whole process down, and every 5 years or so after the age of 25, you start seeing a more and more of a drop as far as being able to maintain certain activities or be able to bounce back from certain types of activities we had in the past, whether it be something as simple as walking down the street or riding a bike. As we age, certain things start to slow down, if not stop entirely. We also see the formation of granulation tissue, Granulation tissue and adhesions, which is going to be scar tissues that will contract over time. This, again, it will be filler tissue. So a lot of times this may not be new tissue, let's say in the muscle and or in cartilage, but actually may be a loose mess of loose connective tissue, which tends to actually contract over time. The collagen fibers start to contract and almost cause a puckering effect. These adhesions a lot of times tend to be very sticky, will adhere themselves to different organs and actually start to cause traction in those areas too. You'll find women that have had C-sections over a long period of time. A lot of times the abdominal wall will adhere itself to the uterus, which causes a lot of painful contractions there as well. You'll find people with deep injuries to certain muscles and or joints. The same thing happens. Scar tissue will start to impede either the contractileness of the actual muscle tissue or actually impinge upon ranges of motion of certain joints. When we get into the joint chapter, we'll actually investigate how well each one of us can move through different ranges of motion. And of course, no surprise to anyone, injuries a lot of times can actually limit how well we can move certain types of joints within their full range of motion. Different types of disorders, typically for the epithelial tissues, and we'll talk about mostly skin cancer for epithelial tissues and burns, tend to be very specific to the organ itself, whereas disorders of connective tissues tend to be more autoimmune. Lupus erythematosus, we'll talk about later on as well. Disorders of the muscular and also the nervous tissues tend to be a little bit more specific, depending on particular tissues, and also whether it's more metabolic or whether it's more dietary, whether it's more behavioral. We'll talk about those later on in different chapters.